There has always been something about sexually transmitted disease that makes people want to place it somewhere else. Blame another country, a particular era, or a particular group of people. The history of how we talk about these diseases is almost inseparable from the history of trying to make them someone else's problem. But the biology doesn't cooperate with that impulse. Most people assume STDs are a product of modernity. Dense cities, large populations moving around, the social changes of the 20th century. And while those conditions absolutely affect transmission rates, they did not create the diseases. The organisms that cause them are old, much older than cities, civilization, and in some cases, older than our species. Proving that is a challenge. STDs primarily affect soft tissue, genitals, mucous membranes, skin. Soft tissue does not survive in the archaeological record. It disappears over centuries and millennia. What survives is bone, and most sexually transmitted infections don't reach bone. At least, not quickly, and not in everyone. So we're already working with a record that is incomplete, and then trying to find the subset of cases severe enough to have left a mark on hard tissue. Scientists call this preservation bias. We are only seeing the fraction of ancient disease that happened to survive into a form we can examine. Which means, when we do find evidence, we know we are seeing the tip of something much larger. And we do find evidence. Treponema pallidum is the bacterium that causes syphilis, and in its late stages, it destroys bone in a very specific and recognizable pattern. Lesions, pitting, a kind of structural damage that is distinct enough that researchers can identify it across thousands of years of decomposition. Those exact patterns have been found on ancient human skeletons going back well before the period most people associate with the disease. There is a long-running debate about syphilis and geography. For a long time, the dominant theory was that Columbus's crew brought it back from the Americas in 1493 and introduced it to Europe, which then experienced a rapid and devastating epidemic in the years immediately following. The timeline seemed to fit. The documented outbreak was real and catastrophic. Armies were being hollowed out by it. Contemporary accounts describe symptoms in clinical detail that match advanced syphilis almost exactly. Then researchers started finding pre-Columbian European skeletons with the same bone damage, and the debate has never fully resolved since. A large 2024 study compiling skeletal data from across Europe confirmed cases that predate 1493 on the connectivity, though other researchers have challenged the methodology and the interpretation of individual specimens. The argument is ongoing, but here is what the argument is actually about. It is a debate about geography and transmission routes, about which population had it first and how it moved. It is not a debate about whether the organism is ancient. On that question, the genetics are fairly clear. The Treponema family of bacteria has been living in and on mammals for an extraordinarily long time. Its closest relatives are found in non-human primates. The branch that leads to Treponema pallidum did not emerge in the 15th century. It evolved over a much longer timescale, in populations of animals that existed long before the age of European exploration. The deeper you go into the genetics of these pathogens, the older everything gets. Herpes simplex virus 2 diverged from its ancestral virus somewhere between 1 and 3 million years ago. That predates Homo sapiens entirely. Our species has only been around for roughly 300,000 years. Whatever HSV2's ancestor was doing before that, it was doing it in hominids that came before us. Researchers believe it may have entered the direct human lineage through contact between different ancestral populations. Populations we now know, from ancient DNA evidence, were not genetically isolated from each other. Early humans interbred with Neanderthals. They interbred with Denisovans. These were not clean separations between species. They were porous boundaries, and what moved across those boundaries included more than just genes. This is actually what you would expect if you think about transmission from a purely biological standpoint. Sex is one of the most efficient pathways that exists for moving a pathogen between hosts. Direct contact between warm mucosal tissue. No requirement to survive outside a body. No need for an intermediate vector. A microorganism that evolved to travel along that route had a serious selective advantage over one that had to survive in open air or in water or in a third-party host. The organisms associated with sexually transmitted infections did not emerge because of anything uniquely modern about human behavior. They emerged because sexual reproduction exists, and wherever sexual reproduction exists, there are organisms that have had millions of years to figure out how to exploit it. This is not a vulnerability specific to humans. It is a consequence of being a sexually reproducing animal. Bacteria and viruses do not distinguish between ancient and modern hosts. They respond to biology, and the relevant biology has not changed. So why does all of this feel like a recent problem? The biggest reason is detection. Ancient people could observe symptoms. They could see lesions and sores. They could notice that certain symptoms seemed to appear after certain kinds of contact, and some of them made that connection explicitly. But without any framework for understanding microbiology, without the ability to identify an organism too small to see, the cause remained invisible. What was visible got interpreted through whatever explanatory system the culture had available. Humoral imbalance, moral transgression, the wrath of something larger than medicine. The Roman physician Galen described genital lesions in the 2nd century. Medieval European physicians documented what they called the Great Pox with enough clinical precision that modern researchers can read those accounts and recognize syphilis. Egyptian medical texts describe genital conditions. The suffering was extensively documented. What was missing was not observation, it was mechanism. The other reason these diseases feel modern is that the consequences of surviving long enough to develop late-stage infections are now more visible. Most ancient people died young, not always violently, but infection, famine, and injury kept life expectancy low. The most severe manifestations of some STDs take years or decades to develop. A person who died at 30 from something unrelated might have been carrying an infection that would have become debilitating at 50. That progression simply never happened. The disease was present. The full course of it often wasn't. There is also a cultural dimension to how the record has been shaped. Diseases associated with sexual behavior were less likely to be written about plainly, less likely to be attributed accurately, more likely to be absorbed into moral narratives than medical ones. A physician in the ancient world writing about a patient's genital condition was navigating social terrain that had nothing to do with diagnosis. That pressure did not make these conditions disappear from the record entirely, but it compressed and distorted what got recorded. The written evidence we have almost certainly represents a fraction of what was actually happening across populations. The stigma that makes these conversations uncomfortable today is not something that developed with modernity. It is as old as the diseases themselves. What makes all of this more than a historical curiosity is that understanding the deep evolutionary history of these pathogens has direct implications for how researchers study and approach them today. The Treponema family has been studied in non-human primates specifically because those related strains help illuminate how the bacterium evolved, what it responds to, and why it has proven so difficult to develop a vaccine against despite being completely treatable with antibiotics once diagnosed. The long relationship between HSV2 and hominid immune systems is part of why the virus is so adept at latency, at hiding from immune response and reactivating. It has had an enormous amount of time to get good at surviving in us. Ancient humans lived with these infections. They suffered from them, tried to treat them, developed social and religious frameworks that attempted to explain them, and passed them forward across generations. The organisms doing the infecting were already ancient when the first cities were built. They were present when humans were living in small bands, moving across landscapes, making contact with other populations, building the deep biological history that we are still, in many ways, only beginning to properly read. The infections did not start with civilization. In several cases, they were there before we were.